So welcome to the current edition of Outstanding Discoveries from CALS, groundbreaking discoveries from CALS. Last Monday, Millard Sussman talked to us about the discovery here of bacterial conjugation and transduction. And today, Nicole Perna from the genetics department, professor of genetics, is going to pick up where he left off and bring us into the present and maybe the future.
Okay, thanks. Is this mic working? Okay, great. I have to confess, when Tom and Dave originally contacted me and asked me to speak in this series, I wasn't sure if I was the old-timer or the newcomer. Then I heard I was paired with Millard, and I figured it out. So I have been around a while, but not as long as Millard. I hope most of you got to see his talk on Monday. It was very interesting, and I'm going to try to pick up where he left off. On Monday, he talked to you about Joshua Lederberg and a number of his groundbreaking discoveries. And I'm going to pick up with, this was my subtitle for my talk, Still Conjugating and Transducing After All These Years. Then I came up with an even longer alternative, which is a tale of the longest genome sequencing project in history, and the impact of sex on genome architecture, and the lingering challenge it poses for the microbiome. So you can choose any of those titles if you would like. So my story is going to pick up with Frederick Blattner, pictured in the middle, who is now an emeritus professor of genetics. And this is my husband and I, over in this other picture, working when we were postdocs, working with Fred Blattner. We're sitting in the conference room over there in what used to be old genetics, down in the basement, which is where the E. coli genome project began and eventually ended. So Fred, this is a very abbreviated timeline of bacterial genomics. It started in the 1970s with sequences of first little parts of bacteriophage. The first sequence Fred collected was, I believe, 24 bases long, a piece of lambda. And then went on to sequence entire phage genomes, which are relatively short. But shortly thereafter, Fred proposed sequencing E. coli, and I'd like to read you the quote, if I can. From his 1983 science paper. Acquisition of total genetic information about a life form adds a new philosophical dimension. In some sense, the rest of biology becomes merely a matter of interpretation. One might wonder, given the total DNA sequence of Escherichia coli and a big enough computer, could one reconstruct what the organism looks like, what it lives on, what it could be poisoned by, how it behaves? Or will a biological uncertainty principle be discovered that would preclude such a development? So it took Fred a little while, from 1983, to get the project up and rolling. First, he had to persuade funding agencies that this was actually doable. The E. coli genome is considerably longer than anything that had been sequenced at that point in time. But he did eventually get funding, the first round of funding. And by just a mere decade later, in 1992, he published the first piece. I should tell you now, the entire E. coli genome is about 4.6 megabases. So this is a very small piece, the first one they published. It was 92 genes. And I highlighted two words there, lambda and MG1655, because I'd like to go back to Lederberg. So we heard from Millard on Monday that lambda was discovered by Esther Lederberg, then Joshua's wife. And the particular E. coli strain that was the first E. coli genome sequenced was a strain called MG1655. Now that is a derivative from Lederberg's lab of a strain he had originally isolated in 1922 from a patient in a nursing home. So the reason Fred and crew chose MG1655 is because that original E. coli K12 strain had been subjected to all sorts of mutagenesis and UV radiation to curative good old lambda, and a variety of other treatments. And MG1655 was the closest we could get to the original E. coli K12 strain. So moving along throughout the 90s, this genome began to come out in bits and pieces, most of them a little bit larger, increasingly larger as it went along, up until 1995, when there was about 1.6 megabases, or roughly a quarter of the complete genome was done at that point in time. But that's a fairly long timeline. And then we'll just skip ahead, and this is not a proportional timeline, to 2018, specifically last night at midnight. There were 9,887 E. coli genomes in GenBank. Why do I emphasize it was at midnight? Because when I checked at 7 p.m., it was one less. So this is a very rapidly changing field these days, compared to the amount of time it took to sequence that first genome. So now I'm going to layer a few more dates onto here to give you a sense of perspective. This is my personal perspective. I joined the Blattner lab in 1996 as a postdoc. Kind of funny. I answered an ad that Fred had put in Science looking for an evolutionary biologist to come compare 3 E. coli genomes to each other. So already, remember, Fred has only sequenced a quarter of the first one, and he's already looking for somebody to analyze 3, which he doesn't even have the money to collect. So I arrived, and I like to think I contributed positively to getting it done in very short order from that point. I'll layer a few more dates, arrows onto here. So this genome took a really long time compared to some other genomes, and it was not the first genome of a free living organism published. That honor went in 1995 to the complete genome sequence of another bacterium, Haemophilus influenzae. It was sequenced by Craig Venter's group at a nonprofit called Tiger in Maryland. So how come they beat us? Well, there's something of a clue right there. I mean, they purposely chose a very small genome, one that didn't contain a lot of repetitive DNA. That made it considerably easier. They also had the advantage of early access to automated DNA sequencing technology, which had been developing in a series of steps in the years that Fred was actively working on the E. coli genome project. I brought a couple of props along. Millard, you're not allowed to answer this, or Dave. Does anybody else in the room know what this is? Not sure if you can see it in this light. This is an autorad from manual sequencing. All right? And the way this works is each 4 lanes on this was once a gel between 2 slabs of glass, and then the radio-labeled nucleotides are what allow us to visualize it on this X-ray film. So each of the 4 lanes corresponds to one of the 4 bases. And to read this DNA sequence, I mean, how do you get from this to a computer where you can actually work with these sequences, right? An army of undergraduates with digitizing pens. I think in the final analysis, there were about 150 undergraduates that were involved with digitizing this sequence. And over a third of the original E. coli genome sequence was sequenced using this technology. Later, we moved to the automated DNA sequencing and redid the whole darn thing. And I have to confess, this is not an autorad from the original E. coli project, but rather from Phi-80, because we just could not wait to throw out the wall of these things we had corresponding to the E. coli genome project. And we lived in fear of somebody asking us to double check the sequence in an area. I want to show you one more thing. Okay, these days we use pretty sophisticated bioinformatics to track progress on a genome project. This is how we track the E. coli K12 MG1655 genome project. Each one of these corresponds to a clone, and then those little things that are taped onto it was every time we would solve another part of the sequence, we would tape one on. There are approximately 20 of these in this pile here, and they used to line my office down there in the basement of genetics. So, I also put on here the yeast genome, because this was the first genome sequence of a eukaryotic organism, and the human genome projects. And you can see these came out in pretty rapid succession. One thing that distinguished the E. coli project and the yeast genome project from one of the two human genome projects was the general approach. The second human genome project was pioneered by Craig Venter of the Haemophilus influenzae, and it used a different approach. So here are the two approaches. So I showed you that sheet. Each one of those corresponded to a subclone. This is the strategy we used over here on the left. So given a target genome, the first thing that happens is we make subclones in some sort of plasmid, or in this example, a bacterial artificial chromosome vector, which could hold maybe 150 kilobases of DNA in any one of those. We make a highly redundant library, and then we have to find a minimum set of those individual clones that will cover that whole region. Having arrived at that, we would take each one of those and then further subdivide that, subclone that yet again, and then yet again, each one of those. And that is incredibly labor intensive. The alternative to this, and the way most genome projects get done these days, is the whole genome shotgun approach. And I chose the simplest figure I could find for it to emphasize how easy it is conceptually. You take all the DNA from an organism, randomly fragment it into little bits, and then sequence those little bits. Now where that becomes complex, over here, we had some idea where the little bits we sequenced belong. Right? Over there, all we know is we have a whole genome worth of sequences. And that is very computationally intensive to put all those bits and pieces together into a larger cohesive unit. So, the Human Genome Consortium, led by Francis Collins, also used a subcloning approach, and so did yeast. And the thing that distinguished those two projects from the E. coli genome project was those were both done by large consortiums. Yeast, it was about 100 different laboratories, each contributing a sequence of a subclone. The Human Genome Project, I don't even know how many laboratories were involved with that. But the E. coli genome, although it took a long time and it cost a lot of money, 150 undergraduates aren't that cheap, not to mention all the other technicians involved. So it cost a lot of money, but it was the longest continuous sequence that was done by a single group at that point in time. And yes, we did finish it, and it was published in 1997. That's a subset of the people that were involved with the project, merited authorship. We had t-shirts printed up, and I searched high and low through my house this morning, hoping I could find mine. I couldn't. We had t-shirts printed up that had 225 names on it of people who had contributed to the project, printed very, very small. I remember the night we completed this. This was, not only were we desperate to complete this project, but we were in a desperate race to beat another group. So there was a group in Japan who was also sequencing an E. coli K12 genome project. And after over a decade of time and millions of dollars invested, we did not want to lose this race. So we were working around the clock. There's a name right there. Oops, I do have a pointer. Jason Greiger. Jason he had a bachelor's degree in math from University of Chicago, and he was our chief editor. So it was his job to take those bits of sequence and figure out how they belong together. He was a bit of an unusual character, so he ate a lot of sunflower seeds. And we were there at 3 a.m., and suddenly there was this rain of sunflower seeds coming down on my head. My desk was right next to his. And Jason jumped up and started doing a jig. And he had finally figured out how to close that final gap in the E. coli genome project. So that was a tremendously exciting night. Then we went home and got some sleep. We can see something of the lasting impact of this. And one way to measure it is how many people have cited this genome paper. And according to the Science Citation Index, as of, again, midnight last night, there were 5,032 citations of this. And we could compare that to Haemophilus influenzae, which was ringing in at 3,993. You can imagine a lot of people cite that Haemophilus paper as the first genome sequence of a free-living organism, so that maybe it'd be fairer to compare it to another genome. So in comparison to Yersinia pestis, the causative agent of the Black Plague, which we sequenced several years later, that has 394 citations to date. So you can see that a lot of people are making use of this E. coli genome project. So what happened with our competitors? Well, eventually we healed the wounds from the rather vigorous competition. We co-published an update to the E. coli genome in 2005, along with a whole bunch of other groups that have been working on E. coli. And here's a picture from 2014 in my office. This is Hiro Mori, who was leading the Japanese group that we were competing with. And I could not find the other picture, but we were holding a piece of that map, which he thought was hysterical. Okay, so I can also see the impact just by looking around on this campus, right? And I chose a subset of people on this campus that are actively working with E. coli that I know have made use of this genome sequence. And many of them I've collaborated with over the years, not all of them. But you know, this gives you a sense. I mean, there's people here from the med school, School of Pharmacy, the engineering, and I kind of weighted it fairly heavily toward CALS, given the subject of this class. I would like to say just a couple of words about Jenny Reed's work, because if you remember back to that quote I read from Fred, he wondered if, given the E. coli genome sequence and a large enough computer, would we be able to predict all these properties of E. coli? Would we know what it could eat? Would we know what could kill it? Well, this is exactly the sort of thing Jenny does with her system scale modeling. So she has a model that incorporates the function of all genes with a known function into a single predictive model. And she can ask, you know, what would happen if I fed the E. coli this much glucose? You know, what would be the growth rate? What if I did it in the absence of oxygen? So this sort of modeling is really getting pretty close to what Fred envisioned in 1983. You can also see, just from looking at some of the topics that these people are studying, just how basic E. coli, how much basic research is done using E. coli. Right? I mean, transcription and translation, replication, RNA polymerase, right? These are things that are broadly conserved across all living organisms. And if you open up a molecular biology text, a lot of what you read there is going to be derived from work done with E. coli K12. So this genome may have taken a long time, and it may have been very painful, but you know, I feel very proud to have had a small part in something that has such a lasting impact. I'm going to show you a couple of figures from the genome paper now, because what I want to do is kind of get back to this topic of bacterial sex, conjugation, and transduction that Millard introduced. So even when we had just one E. coli genome sequence, we could already start to see some hints of this. And I'm going to show you some of the evidence that came screaming out early. I know you can't see a lot on some of these slides, but just bear with me. So this is showing us the base composition along the chromosome. On the first line is the first codon position of all the genes across the entire chromosome. Each line corresponds to the chromosome. Right, and I actually still remember the exact length of it. It's 4, 6, 3, 9, 2, 2, 1 base pairs. It's not that anymore. We've done a couple of sequence corrections, and I have no idea what the number is now, but we were so excited about it then that that number is emblazoned in my head. So as we walk along, and no matter what codon position you're looking at, all codon positions, intergenic regions, what you can see is this base composition, which is just the GC content, as you move through a chromosome, is fluctuating pretty radically. Right? And those fluctuations were a hint that there's some fundamentally different process going on, and it was sprinkled throughout the whole chromosome. I'm going to show you that another way from another figure. This is also from that the original E. coli genome paper, and this is one of 6 pages from a fold-out figure. I shudder to think what the page costs were for that. And what we're seeing here is along the top of each panel are the predicted genes of E. coli, and the ones that we had some insight into the function of are colored, various colors corresponding to different functional groups. The ones that are white are ones that are of unknown function. And at the point we finished this project, it was about 25% of the E. coli genome was still of unknown function. Today, the number is tragically close to 25%. The last count I saw was about 24%. So there's still a lot we don't know about E. coli, even though it's arguably the organism we know the very most about. Down below that, and we'll skip over a couple of lines, there's a series of lines that have these little boxes. Those are actually histograms showing how similar each gene was to the best match in one of these other genomes that had been sequenced to date. And because so few had been done, we had kind of limited comparisons we could make. We compared it to Haemophilus and a handful of other bacteria that were finished by this time, and the yeast genome. And then there's a couple more things on here that you don't need to think about. So what can we see here that tells us something about how much horizontal transfer or bacterial sex is going on? Well, I'm going to call your attention to a couple of different regions. So if you look at some regions, like these ones I've circled in red, you see there are lots of genes in other organisms that are fairly similar, right? And most of the gene functions are known. You can see that over and over again. Here's a particularly well-conserved one. But then there were regions like this that Fred had termed gray holes. As far back as that first segment of the E. coli genome, what are these gray holes? It was harder to predict the genes. We had no idea what they did, and they didn't match anything. So these were, again, indications. And if you look carefully, believe me, I stared at this thing for countless hours. You'll find that this is a very non-random distribution. They're clustered. We even did some statistics on it eventually. And so this is telling us that there are these little pockets of the genome that are fundamentally different. I also want to take this moment to add, we had to develop the software to make this visualization. I mean, this was long enough ago in the field of genomics that you couldn't just go to PubMed and look up, I want to draw a genome, show me a bunch of programs I can download to do it. I mean, it took us several months to design this piece of software just to make this and that circle diagram that you saw before. I still continue to use this piece of software to this day. Okay, here's another thing we could see. Although I showed you the different codon positions, we didn't talk about codon bias. So every bacterial organism, actually every living organism, has a characteristic codon usage. It varies from gene to gene. Highly expressed genes tend to use the most favored codon for any given amino acid. Genes that are expressed at lower levels can get away with a less popular codon. I took this, this is the entire genetic code here with the frequencies, but just to look, here's the 4 codons that all encode proline. And you can see from these percentages that by far the most frequently used one in E. coli is CCG. And there are differences between the other 3 also. So what you can do, given this sort of matrix describing what a typical gene looks like, is you can measure how different is each gene from this sort of average. And that is something, we had several different metrics of it, but the most well-known one is something called the codon adaptation index. And that's actually what this big flare is on this eyeball. So each layer of this is showing a different aspect of the chromosome. Here's the genes on one strand and the other. There's tRNAs, ribosomal RNAs. I forget what that one means. But then here, this big flare is showing us the codon adaptation index for each of those genes. And I think what you can see are there places, and they're highlighted in a slightly different color, where that value is much, much higher than the baseline surrounding it. And if you can make out along this line, these little red-pink flares along that line are genes that had some similarity to bacteriophage genes. And there were no functional prophages in this genome, having been cured of lambda, right? None that we thought were capable of excising. But there were remnants of bacteriophage that had integrated into the chromosome. And I think what you'll see is that many of those flares with an unusual composition correspond to areas with a high concentration of genes that match one or another prophage in the existing sequence databases. However, there are many of those that also, cures a flare, and there's no sign of anything that looks like a phage. So not all of them corresponded to integrated phage. But again, you can see that those flares and the deviations in it are all over the chromosome. So all of these things suggested that those regions of the chromosome that showed an unusual base composition, that showed unusual codon usage, that showed a high proportion of unknown genes, had evolved through some fundamentally different mechanism. We would have had a really hard time arguing it was horizontal transfer mediated by transduction. Maybe we could have argued on the basis of the phage here. But it would have been hard to argue that the other ones had arisen in that way until we had the second E. coli genome, which is where we're headed next. Let's see, by now I joined the lab in 1996. I got my faculty position in 2000. So here we are in the first year of my faculty position. I finally have the second of the 3 genomes I came here to analyze. And I had to sequence it myself with Fred. And what this is I'm showing you here is a comparative map of the 2 genomes. So, and I'll add here again, we had to develop a tool not only to draw this, but to generate the data. I mean, even to compare 2 roughly 5 million base pair long sequences that had huge insertions and deletions relative to each other, we had to develop a new alignment algorithm. And here's the result of it. This sort of solid blue band, so I think I have this on the next slide, this sort of solid blue band is showing you areas that match between the 2 genomes. And where they matched really, really well. They're about 98% identical at the nucleotide level. And that goes all the way around the chromosome, and it's fundamentally the same genes, a different version, slightly different version, same genes in the same order and orientation along the entire chromosome. But then, interspersed among that backbone, as we called it, are regions where the 2 genomes don't match. And those are distributed throughout the whole chromosome. So everywhere you see one of these orange or red blocks in this outer circle is a place where one or the other of the genomes has something totally different. All the cases that are shown in orange are cases where the second genome, which is a pathogen, this is the E. coli 0157H7, associated with undercooked meat and spinach and various other things. So every place that has something that's different than what's in K12 is shown in red, unless there's also something in E. coli K12 that's not found in the 0157H7, in which case it's shown in orange. And I think at a glance, you can see that in the vast majority of these cases, there's something in both genomes. They're totally different sets of genes, but they're in the exact same slot of the genome. And once again, these correspond to this is now showing us something about the base composition along there. And each one of those islands that's inserted into this backbone shows a slightly different compositional signatures than the surrounding areas. So when we tally all this up, what are we left with? I mean, more than 20% of the E. coli 0157H7 genome is not found in E. coli K12. Right? These are 2 things that we call the same species. And yet, 20% of the genome is not homologous. And this starts to build up quite a bit of evidence that what we have is horizontal transfer, bringing in genes from other lineages entirely outside of E. coli into this E. coli genome. And when Miller told you about bacterial conjugation on Monday, he was largely talking about evidence that was based on having two different alleles of the same gene. And horizontal transfer can definitely do that. But what we're seeing here is also a really powerful force for the introduction of novel genes into different members of the species. Okay, a year later, I got my third genome, finally. I didn't call it the third genome because in the meantime, the same Japanese group that had been competing with us to sequence E. coli K12 came out with an E. coli 0157H7. So our E. coli 0157H7 was from a contaminated meat sample from Michigan. Theirs was from contaminated radish sprouts in the Japanese school lunch program. So this was technically the fourth E. coli genome to come out, but it was my third. So what we're seeing here is a Venn diagram. And I hope what you can see, the third one that we added is yet another sort of pathotype. There are E. coli associated with a lot of different types of disease. Most of us immediately think of diarrhea or bloody diarrhea. But there are ones that are urinary tract pathogens also. And so this third one we sequenced was a urinary tract pathogen. And when we build this Venn diagram of the number of genes that are found in all three of them, we are now down to 2,996, which is a mere 39.2% of the total number of genes we saw across all these three species. And each individual organism has a totally unique component, unique as of the count of three, and some of them were larger than others. The urinary tract pathogen had the largest lineage-specific component, and then it also had some genes that were found in common with the bloody diarrhea pathogen. It's 204, and it had some that were in common with K12, but not 0157H7. It's on another 193. So now we're left with, good heavens, what do we mean by a species? If we can have this much variation in a species, this led to a lot of debate about the very nature of bacterial species and whether one could ever have a single phylogenetic tree, a tree of life, that could describe a bacteria if they had this many of the genes coming in from a totally different source. So this sort of work that we were doing, and at the same time people were sequencing other groups of organisms, led to this concept of a pangenome. And what we mean by a pangenome is all the genes found in the population, species, or whatever taxonomic unit you want to talk about. And the general emerging concept was there's a set of core genes that are going to be found in all of them. And then there's a set of variable genes. And if we look at any one organism, we're going to have a little set of variable genes in a core. But if we look across the whole species, the size of that core is not going to change that much. But what changes is the number of very different genes that we see. How much so? Well, here's another way people like to look at it with these graphs. And this could take me a minute to describe to you. So what we're seeing here is a comparison made in this wonderful paper by Marie Touchon. So I was not involved with this work. It was a really amazing piece of work. At this point, there were 20 E. coli genomes. And so if you had just one genome and you asked, how many core genes are there? Well, if there's one genome, all the genes are core. They're found in all of that one strain. That's what that means, right? So the number of genes range from about 4,200 to about 5,600. But as you add additional genomes, and let's say we take any 2 from that set of 20, then we get this distribution. We have now the core is shrunken a little bit, and if we have 3, but you can see it sort of levels out, right? Somewhere around here where we seem to be converging around 2,000 genes that are found in absolutely all E. coli. I haven't done the analysis of the 9,887, but I have done an analysis that goes beyond E. coli to the whole family of enterobacteria that E. coli is a member of. What I can tell you is across the whole family, which is about 500 million years of evolution, so 500 million years of evolutionary time, this core number sinks to about 1,500. Now, when we talk about what happens with the total number of genes that are found in this species, we get a very different story, and that's this graph over here. And as we add additional genomes, you can see as of the point we had 20, the total number of genes, totally novel genes that we were finding is continuing to grow. And there's no sign, well, maybe a slight sign that it's beginning to plateau, but it's certainly not there yet as of 20. You don't have to worry about the other lines. So this is telling us, I mean, well, let's look at this number here. So as of 20 E. coli genomes, we have about 1,700 distinct genes in this species. That is coming very, very close to the number of genes in something like a human genome. Right? So E. coli, as a species, has as many genes as human. Humans don't tend to differ a lot in the number of genes from human to human. E. coli strains certainly do. But when you consider the species as a whole, you could argue that E. coli is just as complex. So does everything work like E. coli? Well, if you ask an E. coli biologist, the answer is, oh, surely it must. However, it's not necessarily true. So what we find is different groups of bacteria are having a different amount of sex. Whether it's through conjugation or through transduction, it varies from organism to organism. So here we might have what we just were looking at there, something called an open pangenome. So it's continuing to grow as we sequence additional strains. And compare that to something called a closed pangenome, where essentially, once you sequence a certain number, you're not finding anything new. And then over here on the right is just another way to look at that sort of data. And here, let me kind of orient you by finding E. coli on here. So E. coli, we can see by the slope of this, this is essentially the number of new genes discovered and normalized by something. So we're still, it's not a huge number, but we're still in the positives there. And compare that to something like Bacillus anthracis, which experiences almost no horizontal gene transfer, no sex. So anthrax, we have a much steeper slope on that. And essentially, there are no new genes discovered as you sequence additional anthrax genomes. You can see, I think, if you look at the slopes of these curves, some of these guys, in fact, a very close relative of Bacillus anthracis, Bacillus cereus, is actually still growing at a more rapid rate than E. coli. So it's not following strictly along taxonomic lines, not like all bacilli are like this. So there's some variation species to species. Okay, now I want to flash ahead sort of to the modern era, the microbiome era. And if you haven't heard the word microbiome, I'm very surprised because it's everywhere these days. So this is in recognition that microbes are everywhere. They're in and on us, all over us, in all natural environments and man-made environments are just covered with microbes. So the microbiome projects that are going on these days are largely directed at finding which bacteria, how many different kinds, how variable are they from, say, one human to another. So it doesn't matter if you live in the same house, doesn't matter if you're the same gender, doesn't matter if it's your offspring. How does that collection of which microbes are present in each environment change? Is it stable over time? What happens if you give somebody an antibiotic? Is the whole microbiome wiped out or only part of it? Does it come back later? There are all sorts of questions, and not just about humans, right? About all these other environments. I saw a fantastic talk today about the microbiome of early land plants, which was quite fascinating. So there's a huge amount of this work being done right now, and I tried to get some estimate of it by taking this figure from PubMed. If you search for a term, it'll show you the distribution of that term across years. So here is now, 2018. Couldn't figure out how to flip this axis. I'm sure there's a way. And it goes back to 1956. There are a few points along here. I find that quite shocking because most people attribute the term to 2001, as I'll show you in a minute. But what you can see is an enormous rise in the number of publications using this term microbiome. And it is continuing to increase. This is now what we're in February. I had to think for a minute, in February of 2018. So this is going to shoot up far beyond 2017. Who knows where the term microbiome came from? Anybody want to guess? Our old buddy, Joshua Lederberg. Not one of his earlier works, but it's from a 2001 Treasury of Words in The Scientist. And he defined this, he defined this in a rather human-specific way. The term is now used in a much broader sense. But it's the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. Once a visionary, always a visionary, right? Sometimes you see this word microbiome used interchangeably with the phrase metagenomics. Does anybody know where the term metagenomics came from? First use of it in the publication goes to Joe Handelsman and Bob Goodman, who were at the time professors in the Department of Plant Pathology here at the University of Wisconsin. Joe left here for Yale, where she did research for a couple of years before joining President Obama's administration as a science advisor. And if you don't know, she's now back here as the director of the Wisconsin Institute for Discovery. And Joe was primarily looking at soil, not humans, but soil, in a search to find new antibiotics, which arguably might be found in soil where the organisms are competing with each other. Perhaps those could be adapted for human use. So although the whole world attributes metagenome as a phrase to Joe, I learned at the genetics retreat last year, she actually credits Fred. So she said one day she was going out to her car, Fred waylaid her, and she was telling him about this project, and he came up with the term metagenome. So there we go. We got both Lederberg and Fred back into this. So once again, there are two basic approaches to surveying microbiomes or metagenomes, and both of them are impacted by the amount of horizontal transfer. Conjugation, transduction, and transformation is the third mechanism by which it happens. Let's take a look at these two methods. Basically, they all start out with all the DNA from a sample, whether it's soil or a swab of somebody's elbow or their mouth or land plants, vascular organs. And then you have two different options. I mean, one is to look at the ribosomal DNA. And the logic behind that is this is a very conserved gene. Right? So I put 16S there, but if you also wanted to include eukaryotes and add another set of primers, it'll pretty much amplify any eukaryotic ribosomal RNA, DNA as well. So you amplify with those conserved primers, and then you can make a figure like this. And I thought about putting this on its own slide, but honestly, it's not any more legible when it's full size. So what this is showing us here is each column is a different sample. Over here, these ones that look kind of similar are all from vertebrate gut. And each of the colors is a different taxonomic group. And if you could read this, and I honestly couldn't read it full size with my glasses on, but I read the figure legend and it told me what those were. Those are different phyla of bacteria. So at the resolution, this is a pretty high taxonomic resolution, right? We're kingdom, phylum, so we're way up at barely specifying what these organisms are. You can already see like radical differences between the gut and over here, I believe that's soil. And then we have various other types of samples here. Okay, so that's one approach. And then you can count up the number of organisms you get in each one of these taxonomic bins and compare across these different samples or across time for, say, a given person. The alternative is to use good old whole genome shotgun again. Once again, we're going to transfer the burden to the computation here. Right? These sequences are relatively easy to deal with because there's huge databases. Like almost every known species has an existing ribosomal DNA sequence that you can compare against. But whole genome, You're kind of working in the dark. So you take all that DNA, and there may be lots of different genomes. In this example, I think there are 4 different colors here, 4 different types of bacterial genomes. You fragment the whole kit and caboodle into pieces and sequence it, and then rely on your bioinformatics to stitch it all back together in some sort of cohesive way. Turns out that's hard. Right? So, and one of the reasons why it's hard is, because I have no way of telling that this assembled orange contig or contiguous stretch of DNA comes from the same organism as this one. There's nothing that says, hey, I'm from the orange species. They're nicely colorized here. If they came out of the computer that way, you'd be like, okay, I'm done. I can go home. Right? But they don't. What you get are these 4 collections of things, and you have to figure out how many things are in there. So both of these things are implications of horizontal transfer. So on this side with the 16S, it sort of determines who's present, but with pretty poor taxonomic resolution. A best case scenario is maybe you can get to the genus, in some cases down to the species level. But I just spent this whole talk telling you that if you tell me you have E. coli present, I can't tell you whether it's a absolutely harmless laboratory strain or whether it's a deadly pathogen. So what good does it do to me to know that there's E. coli in soil and E. coli in human? So there are other applications. We could clearly see differences between the samples, and if they're different at that high taxonomic level, they're surely different at a low taxonomic level. But it's hard to know quite what to make of it. Over here, one of the challenges here in figuring out which pieces belong together is this horizontal gene transfer. Even though we may have one exemplar of an orange genome, we don't know whether this orange genome is going to be exactly the same as the other orange genome, or is it going to have those islands sprinkled throughout it? And maybe the reason that hole is there is because we don't know what we're looking for. Right? So the amount of horizontal transfer, amount of conjugation, transduction, and transformation is going to impact how easy it is to piece this together. Another thing that might immediately occur to you that will matter is just how many different organisms are in your sample. Right? If you've got a metagenome and there's only 3 different organisms in there, it's going to be a lot easier to piece together than if you have a metagenome that has thousands of different organisms in there. Turns out that's more common. Aren't that many environments that are that uniform. So, but the nice thing about this is we don't just get the sequence of a piece of the ribosome, which is going to look like a piece of the ribosome. Right? If we do this, we can see all these different genes and their potential functions. And this sort of tells us a little bit more about what the microbes are doing in that environment, but it does not tell us who's doing what, necessarily. All right, so I'm going to summarize here. So the first E. coli genome project was visionary. It was challenging. It was expensive, but it's had lasting impact. Bacterial genomics very quickly became easier, and this was a result of both those advances in automated DNA sequencing and advances in bioinformatics, so we're not running around with a big piece of graph paper and tape anymore. Thank goodness. Horizontal gene transfer has shaped the architecture and content of individual genomes. The extent to which depends on which group of organisms you're talking about. That's another thing that makes that metagenomics challenging, right? Some of them might be easier to put together because they have lower rates of horizontal transfer than others. So there's a bias inherently in your ability to access that information. So bacterial species exhibit this tremendous diversity in content, and then that in turn translates into challenges to interpret the very simplest ribosomal RNA-based microbiome studies and the more complex whole genome shotgun studies. And with that, I'll say thank you for coming and thank the hundreds of people that contributed to the projects I talked about.
Thank you for talking about it. Questions? When people originally set aside a group of cell lines that they call E. coli, what did they all have in common? What makes us think that they belong to us?
Mhm. Yeah, so bacterial species are a really squishy subject, right? So prior to DNA sequencing, it was defined by biochemical phenotypes, right? It can ferment lactose, or it can't ferment lactose, it can do sucrose, not lactose, yes, galactitol, or, and so based on the overall similarity in its metabolic profile, it would make the best determination you could. In the era of sequencing, we now have a kind of operational definition that's based on that 16S ribosomal RNA sequence, that sequences that are 97% or more identical are a species.
That's an arbitrary definition, isn't it?
Why, yes, it is. I have had many a discussion, we'll call it, instead of an argument, many a discussion with bacterial taxonomists, and the sort of bottom line is it's basically a practical definition. We don't have something like a biological species concept for bacteria. And you might think you could do it based on sex, right? Based on which other organisms they're exchanging DNA with. But it turns out they can exchange DNA over fairly long distance in the phylogenetic tree. And although it might be more common within a species, it depends what species you're talking about. So there's no one universal definition that's going to work. These days, there aren't that many classical taxonomists around anymore.
Why?
Because we haven't been training people in that area for a lot of years.
Why?
Probably because nobody knows how to interpret it. It's also just not that sexy of a field. There's not legions of young people who are saying, I really want to be a taxonomist.
I think what we've done is to teach biology as though it didn't involve math. And we haven't had people who could walk both sides of the street, computational science and genetics.
I agree wholeheartedly. I am really happy that I'm starting to see that shift. We increasingly have an increasing number of graduate students in the Department of Genetics who come in with skills in both or are ready to learn the skills in the field that they didn't previously study. And I think that's going to be wonderful.
Did John Devereaux play a role in the early software development?
Yeah, good old GCG was a piece of software.
I think I taught him 40 years ago when he was a taxonomist.
Yeah. He made the right move getting from taxonomy into bioinformatics, for sure.
I think before that he got his degree in history as an undergraduate.
There's also, another interesting trend these days is to move away from the phenotype-based taxonomy altogether and move beyond the just 16S to take into account the entire genome. Since we don't have a fundamental biological concept for what is a species, there are a lot of people pushing for an operational definition that is based strictly on genome similarity, average nucleotide identity throughout the whole chromosome, that sort of thing. That might give us taxonomic bins that make more logical sense, but nobody's going to know what they are. I study bin 0165328. Nobody's going to say, oh, E. coli. So, there's pros and cons to both.
I think it's an impressive fact that the sequencing of coli began here and ended here.
Yeah, it is. There were a lot of people along the way that did not think it would get done, including the funding agencies who had to be talked into yet another supplement in order to get it through. Oh, I don't think we ever had the heart to tally it, but Guy Plunkett, who is working on the project almost throughout it, estimated it would come in somewhere around 10 million. That does also include, though, purchasing those automated DNA sequencing machines circa 1995.
So not as bad as the human genome, which cost
Right, but it's much smaller.
I want to bring this back to a very lay level regarding public health and E. coli. This is very, and I know that this was not the intention of all of you researching this, but that whole realm of we don't know, and we don't know how, even despite all this incredible work, huge number of hours, costliness of the research you all did, the threat of E. coli, public health threat, and dire consequences, possibly that sometimes result in death, as you well know. Does it frighten people who are continuing to try to work in ways that they can in the public health arena about E. coli outbreaks, future ones?
Yeah, I mean, I am finally starting to see some shift in both in the public health agencies, I won't name them, toward a more genomic-based surveillance system. The problem all along has been tracking based on a very small number of genes or markers.
Okay.
And let's say we are tracking E. coli 0157H7 based on one gene. As soon as a strain loses that gene, it's no longer detectable by that method. And when we first sequenced this genome, the standard for tracking E. coli 0157H7, which is one of the more deadly of the E. coli pathogens, was restriction fragment length polymorphisms and pulse field gel electrophoresis. The problem with that is there were only 30 bands. It was really hard to tell the difference between this, is this one exactly the same size as that one? If they're done in two different labs, they might look totally different and be the same. They could be the same size and still have totally different content in them. It was very, very non-specific. Another thing you could do would be to track based on the virulence factors. And I think I forgot to say that, but those islands that differentiate the strains from each other tend to contain genes that are involved with niche-specific phenotypes. Like 0157H7 is a deadly pathogen. All the virulence factors are in those islands. Now, I wouldn't want to base chasing it through an epidemic on any one locus, because again, that single gene might be mutated in one lineage. But if you look at the sum total, all 1,500 genes that are found in that that strain, that group of strains, then you could start to do a more rigorous surveillance. Miller.
When you look at one of these populations, wild-type population of microorganisms, how many of them have plasmids in them? How many viruses do you find mixed in?
Varies quite a bit. So the ones I talked about today, I showed you only one circular chromosome. 0157H7 actually has two plasmids, and some of the virulence factors are on the larger of the two plasmids. It's about 100 kilobases, and then it has a little tiny 3.5 kilobase one. I sequenced the genome of a plant pathogenic enterobacteria, though, that has, depending on where you draw the line between a plasmid and a chromosome, it either has 29 chromosomes or a chromosome and 28 plasmids. That was quite a surprise because we couldn't figure out why can't we get this to assemble into one big circular chromosome? Well, because it's not. So there is variation in genome structure out there and in a number of additional replicons. Varies within species, it varies between species. Viruses. Let's see. E. coli K12 had 9 cryptic prophage in it. I think E. coli, or the strain of E. coli 0157H7 we sequenced, I should say, the strain we sequenced had closer to 25. So it varies, again, within and between species on the number of intact or remnants of bacteriophage you find.
How about horizontal transfer between prokaryotes and eukaryotes?
There are increasingly persuasive examples of that. Many of the early examples kind of got poo-pooed as potential contamination. Right? Anytime you do something like sequence a genome project, You know, there's often something in there that doesn't belong in the genome you're sequencing. But there is increasing evidence of it, and in our own department, Chris Hittinger has, now that he's sequencing quite a few wild yeast, has turned up something that looks very, very much like a bacterial siderophore, an iron scavenging whole operon. You know, these are big. There's about 40 kilobases worth of genes involved with making this thing, sitting right smack in the middle of one of the yeast. And it matches best in the whole wide world of known proteins to an enterobacteria called Serratia. How it got there, I don't know, but you know, Chris even has evidence that it's expressed.
So there's still room for research in this area?
Yes, there is quite a bit of room for research in this area. And one of the really pressing areas right now is this metagenomic shotgun sequencing. You know, people are really struggling to get those bits and pieces connected up together. And one thing, once again, we're going to have a technological revolution that makes it a lot easier. So most of these genomes were sequenced with individual sequences somewhere on the order of 25 to 500 base pairs long. But now there's long-read sequencing. And we did a genome this past summer using Oxford nanopore sequencing, which is essentially squeezes a single molecule out through a pore and measures conductivity. And we had one single sequence that was 165 kilobases. So compared to trying to knit together 25 base pair fragments and find where they overlap with each other, you know, that's an enormous advantage. They weren't all that size, but you know, the technology is moving in that direction.
Thank you very much.
Thank you.
Thanks, Nicole.